Lecture 3 Molecular Genetics Part 1 The structure of nucleic acids. The primary structure of DNA, secondary structures of DNA, denaturation and renaturation of DNA, the tertiary structure of DNA, RNA molecules, central dogma of molecular biology. Question 3.1 The structure of nucleic acids. History of the study of the structure of nucleic acids. 1868. Friedrich Miescher isolated nuclei and identified phosphorus-containing substance, which was called nuclein. Now it is called nucleoprotein. 1872. Friedrich Miescher found that heads of semen sperm cells contain acid substance. Now it is called nucleic acid. 1944. Avery, MacLeod, and McCarthy identified deoxyribonucleic acid as the carrier of genetic information. 1948. Erwin Chagov, with colleagues, discovered rules of base composition of DNA molecules. 1953. James Watson and Francis Crick created the model of the DNA structure. 1961-1965. Marshall Nirenberg determined the genetic code. 2003, the human genome was sequenced. The nature of genetic material. The idea that genes are made of nucleic acids was not widely accepted until after 1950. In 1928, Griffith demonstrated transformation in bacteria Streptococcus pneumoniae. One capsulated strain produced colonies that looked smooth, big S, and killed mice. Another strain without capsule produced colonies that looked rough, big R, and was not virulent. Heat-killed S bacteria did not produce infection. When dead smooth cells and rough cells were mixed, bacterial transformation occurred. The injected mixture killed mice. Griffiths concluded that in the presence of the dead S bacteria, some of the living R bacteria had been transformed into virulent S strain organisms. This kind of change of trait in bacteria is called a bacterial transformation. In 1944, Avery, MacLeod, and McCarthy reported the nature of the transforming substance. They used enzymes to separately destroy protein, RNA, or DNA in heat-killed S cells. They demonstrated that the only enzymes that destroyed the transforming ability were enzymes that destroyed DNA, called DNases. They concluded that DNA is responsible for transformation in bacteria and DNA is the genetic material. The primary structure of DNA. Nucleic acids are complex molecules composed of carbon, hydrogen, oxygen, phosphorus, and nitrogen. Nucleic acids are polymeric molecules made up of many repeating units linked together. The building blocks or monomers of nucleic acids are nucleotides. It is useful to consider the structure of DNA at three levels of increasing complexity, known as the primary, secondary, and tertiary structures of DNA. The primary structure of DNA refers to its nucleotide structure and how the nucleotides are joined together. The secondary structure refers to DNA's stable three-dimensional configuration, the helical structure worked out by Watson and Crick. Tertiary structures of DNA are the complex packing arrangements of double-stranded DNA in eukaryotic chromatin in bacterial nucleoids. The nucleotide structure. Nucleotides are composed of a 5-carbon sugar to which one or more phosphate groups and a nitrogen-containing base are attached. In the case of the nucleotides in RNA, the sugar is ribose. In the case of the nucleotides in DNA, the sugar is deoxyribose. Pentose sugars have 5 carbon atoms, numbered 1', 2', 3', and so forth. In the picture to the left, we can find a chemical structure of ribose. 1', 2', 3', 4', and 5' carbon atoms. 1, 2, 3, 4 hydroxyl groups are attached to the carbon atoms. Deoxyribose doesn't contain an oxygen atom at the 2' carbon atom. The second component of a nucleotide is its heterocyclic nitrogenous base, which may be of two types, a purine or a pyrimidine, or pyrimidine in another pronunciation. Each purine consists of a 6-sided ring attached to a 5-sided ring. In each ring, we can find carbon and nitrogen atoms. What is the difference between adenine and guanine? They differ in the positions of their double bonds and in the groups attached to the 6-sided ring. For example, adenine has amino group that is attached to the 6th atom of carbon. Pyrimidine nitrogen-containing bases. Pyrimidine consists of a 6-sided ring, hence its structure is simpler than the structure of purine. There are 3 main pyrimidine nitrogen-containing bases: cytosine, uracil, and thymine. Cytosine is present in both DNA and RNA. Thymine is restricted to DNA, and uracil is found only in RNA. One exception exists. Thymine can be found in tRNA, or transfer RNA. The 3 pyrimidines differ in the groups or atoms attached to the carbon atoms of the ring and in the number of double bonds in the ring. We can compare thymine and uracil. Thymine has additional methyl group that is attached to the 5th carbon atom. Hence, if this methyl group will be removed, thymine will transform into uracil. If methyl group will be added to uracil, it will transform into thymine. In the left picture, we can find the main structure of the nucleotide. It consists of 3 parts: purine or pyrimidine base that is attached to the 1st carbon atom, 1' C, and phosphate group that is attached to the 5th carbon atom, 5' C. To the right, we can find that if nitrogen base is attached to the ribose or deoxyribose, such chemical compound is called nucleoside. If phosphate group is added to the nucleoside, such compound is called nucleoside monophosphate. If 2 phosphate groups are added to a nucleoside, such substance is called nucleoside diphosphate. If 3 phosphate groups are added to nucleoside, nucleoside triphosphate is formed. The wavy line represents a high-energy bond. We remember that ribonucleotide, ATP that contains ribose, stores energy. Now pay attention to the nucleotide nomenclature. Base adenine, nucleoside adenosine, nucleotide adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. If nucleoside contains deoxyribose, we use terms deoxyadenosine, deoxyadenosine monophosphate, and so on. Abbreviations AMP, ADP, ATP for ribonucleotides, and DAMP, DADP, DATP for deoxyribonucleotides. Base guanine, nucleoside guanosine. Base cytosine, nucleoside is called cytidine or cytidine. Base thymine, nucleoside is called thymidine. Base uracil, nucleoside is called uridine. The nucleotides are covalently linked together in a chain through the sugars and phosphates, which thus form a backbone of alternating sugar-phosphate. Pay attention to the picture to the right. Deoxyribose, phosphate group, the next deoxyribose, the next phosphate group. They form a backbone. The way in which the nucleotide subunits are aligned together gives a DNA strand a chemical polarity. DNA strand has a 5' phosphate on one terminus and the 3' hydroxyl on the other. This polarity in a DNA chain is indicated by referring to one end as the 3' end and the other as the 5' end. Such polarity is of great importance because it determines direction of reading of hereditary information. It is read from 5' end to the 3' end, in one direction only. The nucleotides are linked together covalently by phosphodiester bonds, i.e. COPOC, through the 3' hydroxyl OH group of one sugar and the 5' phosphate P of the next. Thus, each polynucleotide strand has a chemical polarity, that is, its two ends are chemically different. The 3' end carries an unlinked OH group attached to the 3' position on the sugar ring. The 5' end carries a free phosphate group attached to the 5' position on the sugar ring. By convention, the sequence of bases is usually expressed in the 5' to 3' direction. Thus, the base sequence GTCCAT refers to the sequence 5' PG, PT, PC, PC, PA, PT, OH, 3', where the phosphates are indicated by small p. Note that this is not the same molecule as that represented by the reverse sequence 5' PT, PA, PC, PC, PT, PG, OH, 3', or TACCTG. Two sequences, GTCCAT and TACCTG, are quite different. Secondary structures of DNA. Primary structure of DNA means nucleotide structure and how these nucleotides are linked together. They form one DNA strand. Secondary structure of DNA includes two DNA strands that are joined together and that form DNA double helix. Chagov's rules. Erwin Chagov and his colleagues in the late 1940s found that the base composition of DNA generally varies from one species to another. DNA specimens isolated from different tissues of the same species have the same base composition. The base composition of DNA in a given species does not change with an organism's age, nutritional state, or change in environment. And the amount of adenine equaled that of thymine, and the amount of guanine equaled that of cytosine. A equals T and G equals C. It follows that there is always an equal proportion of the purine A plus G and pyrimidine T plus C bases. But Chagov was unable to realize the meaning of his work. A DNA molecule consists of two long polynucleotide chains composed of 4 types of nucleotide subunits. Each of these chains is known as a DNA chain or a DNA strand. Hydrogen bonds between the base portions of the nucleotides hold the two chains together. Adenine A always pairs with thymine T and guanine G with cytosine C, complementary base pairing. 2 hydrogen bonds form between A and T, while 3 form between G and C. Pay attention that in the picture to the right, between A and T, only 2 possible hydrogen bonds can be produced, because distance between N and H, O and H, is very small. Between this oxygen and this hydrogen, distance is very large. Hydrogen bond cannot form. And between cytosine and guanine, 3 hydrogen bonds can be produced. Distance between appropriate atoms is very small. The bases can pair in this way only if the 2 polynucleotide chains that contain them are antiparallel to each other. Pay attention to the picture to the left. Arrows indicate polarities. In this picture also we can find why 2 hydrogen bonds are formed between T and A and 3 hydrogen bonds are formed between C and G. In 1947, Astbury began studying the structure of DNA by using X-ray diffraction analysis, but his diffraction pictures did not provide enough resolution to reveal the structure. In 1952, Rosalind Franklin received DNA Röntgenogram of high quality. She worked in the laboratory headed by Maurice Wilkins. Cross-like structure in the center of this photo proves the DNA is composed of the polymer wound into a helix. 2 large dark structures correspond the distance of 3.4 angstroms thickness of one nucleotide. James Watson and Francis Crick found that a possible structure of DNA was one in which two helices coiled around one another, a double helix, with the sugar phosphate backbones on the outside and the bases on the inside. The observation that DNA was double-stranded was of crucial significance and provided one of the major clues that led to the Watson-Crick structure of DNA. Only when this model was proposed did DNA's potential for replication and information encoding become apparent. In this picture, we can see James Watson and Francis Crick near the model of the DNA. The three-dimensional structure of DNA, the double helix, arises from the chemical and structural features of its two polynucleotide chains. All the bases are on the inside of the double helix, and the sugar phosphate backbones are on the outside. Why? Because bases are hydrophobic, do not like water, and the sugar phosphate backbone is hydrophilic, likes water, contacts with water. The second force that holds the two DNA strands together is the interaction between the stacked base pairs. These stacking interactions contribute to the stability of the DNA molecule by excluding water molecules from the spaces between the base pairs. Pay attention that bases in this picture are parallel. Two strands are antiparallel to each other, which means that the 5' end of one strand is opposite the 3' end of the second. DNA is a right-handed helix. Each turn of DNA is made up of 10.4 nucleotide pairs, and the center-to-center distance between adjacent nucleotide pairs is 3.4 angstrom. The coiling of the two strands around each other creates two grooves in the double helix. The wider groove is called the major groove. It is indicated by number 1 in our picture. And the smaller is the minor groove, number 2. Diameter of DNA is 20 angstrom, or 2 nanometer. Francis Crick, James Watson, and Maurice Wilkins were awarded Nobel Prize in 1962. The secondary structure of DNA that Watson and Crick described is termed the bDNA structure. The bDNA structure is the most stable configuration for a random sequence of nucleotides under physiological conditions, when plenty of water surrounds the molecule and there is no unusual base sequence in the DNA. It is the predominant structure in the cell. The aDNA structure exists when less water is present. It is shorter and wider than bDNA. aDNA also is right-handed as bDNA. zDNA forms elongated and narrow left-handed helix. In this form, the sugar phosphate backbones zigzag back and forth, giving rise to the name zDNA for zigzag. It can arise when stretches of alternating C and G sequences are present and require very salt concentrations to become stable. The DNA can be stabilized in physiologically normal conditions if methyl groups are added to the cytosines. DNA has very important property. It can denaturate and renaturate. The hydrogen bonds can be broken and the DNA strands separated when the DNA molecule is heated in water, a process called DNA melting or DNA denaturation. It starts at approximately 68 degrees of centigrade. Since a GC base pair has 3 hydrogen bonds to every 2 in an AT base pair, the higher the GC content in a given molecule of DNA, the higher the temperature required to denature it. The denaturation of DNA by heating is reversible. If single-stranded DNA is slowly cooled, 100 degrees, 99, 98, 97, and so on, single strands will collide and hydrogen bonds will again form between complementary base pairs, producing double-stranded DNA. This reaction is called renaturation, annealing or reannealing. The tertiary structure of DNA. The tertiary structure of DNA refers to higher order folding that allows very long DNA molecules to be packed into the confined space of a cell. In this picture to the right, we can find that in bacteria, a chromosome is highly folded into a series of twisted loops that are maintained by proteins. In bacteria and in higher organisms, DNA can be supercoiled. Supercoiling, it is the coiling of a coil, occurs when the DNA helix is overwound or underwound. Supercoiling relies on topoisomerases, enzymes that add or remove rotations from the DNA helix by temporarily breaking the nucleotide strands, rotating the ends around each other, and then rejoining the broken ends. Eukaryotic DNA is bound to histone and non-histone chromosomal proteins and forms a chromatin. Its structure was described earlier. RNA molecules have different tertiary structures. RNA consists of ribonucleotides. It has the bases adenine, guanine, cytosine, and uracil. RNA is usually a single-stranded nucleotide chain, but some viruses have double-stranded RNA. Informational RNAs or messenger RNAs, mRNAs, pass information from DNA to protein. They have different three-dimensional structure. Ribosomal RNAs or rRNAs are the major components of ribosomes. They are bound to ribosomal proteins and form large and small subunits of ribosomes. Transfer RNAs, tRNAs, are responsible for bringing the correct amino acid to the mRNA in the process of translation. They have clever leaf structure. The D-arm, it has dihydrouridine residues. The TPC arm, it has minor bases thymine and pseudouracil. And the anticodon arm with the anticodon loop, which contains three nucleotides making up the anticodon, which pairs with the corresponding codon on mRNA to ensure that the amino acids link in the correct order. Three-dimensional structure of tRNA has the form of a twisted L. Central dogma of molecular biology. There are three major pathways of information flow in the cell. In replication, information passes from one DNA molecule to other DNA molecules. In transcription, information passes from DNA to RNA. And in translation, information passes from RNA to protein. The flow of genetic information in cells is therefore from DNA to RNA to protein. This principle is termed the central dogma of molecular biology. Francis Crick, 1958. A central dogma was updated because in some viruses information is transferred from RNA to DNA in the process that is called reverse transcription, or to another RNA molecule in the process that is called RNA replication. How does DNA perform its functions? In this table, we can find DNA function and a cellular process which provides DNA functioning. The first function, information storage, is provided by DNA repair. The second, transfer of information, is provided by DNA replication, synthesis of new DNA molecules. The third, regulation of cellular processes, is provided by transcription and translation. These processes will be discussed in the following lectures.